So, hello, and welcome to Bio 211, your Anatomy and Physiology 1. We're looking at Chapter 3, and we're looking at actually how we pack DNA into a cell. And you'll notice it says here that it's a dividing cell. Okay, so let's go here first. Normal, active transcription and translation requires that chromatin be in a specific shape, which means it's unfurled, which means it's not wound around these things we're going to call histones in a moment, right? And that's important because if you understand that, it means that when you're looking at a cell, you never see chromosomes, okay? It's only when a cell is undergoing division, meaning we are replicated and we are now separating the numbers of chromosomes from one another, that you would actually see them condensed. Okay? So with that in mind, I want to show you that there's the chromatin right there, and what we do is we wrap it around histones to create a nucleosome. And that nucleosome is then packed, and I don't like the vocabulary they're using here. They call it a chromatin fiber. I would probably call it a chromosome fiber, but I see why they're calling it chromatin. It's individual chromatin molecules of genes as a double helix wrapped around a nucleosome. Okay? And then what happens, of course, is you pack it together into the chromosomal arms, of which you have a centromere here in the middle, and each of the arms is now named a chromatid. Now that's a lot of chroma something or others, right? So you got to figure out a way to keep it separate for yourself. So the way I do it is this, is that if it's a non-dividing cell, it's chromatin. If it's a dividing cell, it's a chromosome with chromatid arms. Because I know I have those, it means I know that I have nucleosomes, and those nucleosomes are made up of histone proteins. Okay? So hopefully that helps, because the way your textbook explains it to you, it's a little odd. Okay? Overview of gene expression. Well, we need to make proteins, right? And of your 23,000 genes inside of your cells, you're going to transcribe a gene. It's going to be translated at some other location, and that translation is going to require ribosomes. And you remember we have two parts to our ribosomes. So that means we have to break down first the step of transcription, the translocation, and then two, the translation of whatever that information was from the gene. And pay attention to the color code changes here. So it's purple for the double-stranded DNA. It becomes red for the single-stranded RNA structure. And that single-stranded RNA structure now interacts with our complete formed ribosome with a large subunit and a small subunit. Okay? Maybe that's actually how you have to learn it. I mean, you want to keep it that simple first. You always want to keep it simple first. You don't want to apply all the details your first pass through. So even though it sounds like I'm making fun of this, what I really like about it is this. Gene expression has two major parts to it. One, there's a transcription part to it. The second, there's a translation part to it. The more nuanced thinker is going to be thinking to themselves, okay, I see the transcription piece, but it's still inside the nucleus here. So therefore, translation can happen at the nuclear envelope, or it can happen in the cytoplasm, or it can happen wherever that ribosome is. That's the way I'm hoping you're developing your thinking processes. So, how do we transcribe? Well, up here, inside your textbook, you see inside this cartoon as well, you have a promoter region which says, yes, transcribe. Okay, and then the gene is going to be transcribed using this thing here in yellow. I call it candy corn inside class, but if it's not yellow inside your book or whatever, you get the idea. It's an RNA polymerase. It's an enzyme. That enzyme makes a copy of one of the strands, upper strand or lower strand, of DNA. Okay? And now what that means is you have an intermediate of information. Here's that red ribbon. That red ribbon now has the A's, the U's, the G's, and the C's that when combined in three nucleotide groups, you now have the information to make, or I should say, information so that we bring amino acids to a unique location where they can undergo dehydration synthesis. But before we do that, we have to cut out non-coding sequences. Okay? So let's go like this. And here, I should probably change colors because everything is already red, right? So let's go here. So here, your textbook has said that this entire strip here is non-coding. That does not mean it's not important. It's beyond the scope of this class, though. It just means you need to take this piece out. And to take this piece out, you bring the ends together and you cut it. Small nuclear ribonuclear protein. Okay, they're also called SNRPs, whatever. Makes no difference. You cut them, you readjoin these things, and now what you've done is you've gone from pre-mRNA, and because you've cut out the introns, now you have only exons, and therefore you have mRNA, where M stands for messenger. Okay, so this is the instruction for us to assemble amino acids in the correct order, dependent upon the 3 nucleotides in order for each of these things. That is called a codon. You'll see how it's used in a moment. Okay, so that's the transcription piece. So that's part 1, done. Now, that happened inside the nucleus, so therefore it's going to either be translated at the nuclear envelope, or at the rough endoplasmic reticulum, or the cytosol of the cell. You'll see your textbook doesn't really care when they're teaching you the basic idea. They just say, look, for us to begin translation, we need 3 pieces of materials to be located in the same area at the same time. 1, we need a small subunit of a ribosome. 2, we need mRNA. Okay? And 3, we need an initiator tRNA. And that initiator tRNA always has to be methionine for this class. And you'll see the reason why as the start codon here. I don't know why they represent it this way. Should be AUG. Okay? Which means the anticodon sequence from the tRNA is going to be UAC. And we call this complementary base pairing. This is the mechanism by which your A's bind to T's, T's bind to C's. Did you hear that? That's not right. A's bind to T's, G's bind to C's. The number of people who get that wrong is insane. You shouldn't be getting that wrong at this stage in your careers. Okay? But you'll see that if this is the case for our methionine, take a look at what happens when the next amino acid comes in here. We have another anticodon binding the codon sequence here, and now we actually have amino acids adjacent to one another. Because the amino acids are adjacent to one another, they undergo a dehydration synthesis reaction to create a peptide bond. Okay? And this continues. It continues and it continues until we get to the stop codon. And the stop codon says, okay, enough is enough. You stop bringing in more tRNA. You slice that off, and now we are in our primary strand of amino acids. This will then fold into its secondary structure, and then it will assume its functional shape in its tertiary structure, regardless of what the protein is. Okay? Does it require complex or it doesn't, right? For it to be functional, it has to assume its own tertiary structure. Okay? So go through this a couple of times. Make sure you understand. Notice I'm not talking about P sites and A sites and all that sort of crap. I'm making sure you understand what does a tRNA do. When did that large subunit actually come into our story? Notice I didn't mention it, but for this thing to move forward, we started it with these 3 pieces. Now the large subunit comes in, and then we start processing. Okay, until we get to that stop codon here. Okay, cool. Let's get rid of the cartoons. Let's move on. We want to now think about, if we're making proteins, what's going to happen inside of a cell. Okay, and your text, for whatever reason, just goes immediately to cell division. Somehow it forgets that between making proteins and becoming a cell, you actually did things like grow. Okay, and I want to introduce this idea by simply saying that cell growth, okay, they say cell cycle is a sequence of events in which a body cell duplicates its contents and divides in 2, which is completely true. Okay, completely true. But now think about it like this. You're a cell. You just became a new cell. You got a nucleus. You got to do your job, whatever your job is. Let's imagine it's, I don't know, lining a blood vessel. So this is a simple squamous epithelial cell, and it's going to grow. That's the most important thing, right? It's going to grow. It's going to take up materials. It's going to get slightly bigger than it was. And then at one point in time, it's going to be like, okay, enough is enough. I know now that I have to make 2 of me so I can replace myself. And that's actually what's going to happen. So we're going to go with something like this. And you're going to end up then with 2 smaller cells. Okay? And it doesn't always happen like this. It could be an endothelium and this guy gets killed. Okay? That means the one that's next to it has to grow. It means it has to undergo mitosis rapidly to keep the collagen inside the endothelium covered. But that's the point, is it has to have a series of mechanisms in place where it can duplicate itself. Okay? And that means it has to take 23 pair of chromosomes and make copies of that. So at the end of it, you end up with 46 pair. Okay? Now, because these are somatic cells, we call this a diploid cell. Okay? If we're talking about meiotic cells, those would be haploid cells, where we're going to actually reduce the total number of chromosomes so that if you started off with 23 pair, you end up with 23 chromosomes at the end. So how does it happen? Okay? Well, let's take a look at the cartoon here. Let's get rid of this first. Okay? In purple here is that normal growth period I was telling you about, where you have growth G0, okay, is going, the cell, it's non-dividing. No, G0 is if I say it's a neuron, okay, and that neuron would be, it's non-dividing. That's what you get. The G1 phase here, these are metabolically active cells, are undergoing everything they're supposed to be doing, and at the same time getting ready to replicate the genetic material. What happens here in S phase is essentially all of this stops, and you focus all of the energy on using DNA polymerase, Okay, and that DNA polymerase is now making a copy of all 23 pair of chromosomes. Now I want to make sure something's perfectly clear. It's not one copy of DNA polymerase. It's millions of copies of DNA polymerase that are running around making segments. They're being spliced together. And lo and behold, at the end of it, if we started off with 23 pair here, we now have 46 pair here. And now we need to figure out what we're going to do with those 46 pair of chromosomes. Well, after we've replicated all of this, the signal goes out to the cell like, hey, full steam ahead. And now what's going to happen is the cell is going to continue to grow. And we're now going to make the centrosome replicated. And these will be the tools we'll use to separate the numbers of chromosomes from one another. And this means we have to go back to what is a centrosome. Do you guys remember the centrosome? We used the centrosome to make microtubules. Okay, that's important. Because once we've actually finished that, we're going to actually enter what we call mitosis. So notice this is all interphase. Most of your cell spends its life in interphase. It's only prophase, metaphase, anaphase, and telophase that are unique because what we're doing is we're separating the 23 pair of chromosomes and the other 23 pair of chromosomes so that you end up with 2 functional cells. So you have 46 pair of chromosomes here. By the time we finish mitosis, you have 2 cells with 23 pair of chromosomes. So what's going on? Get rid of the cartoon piece here. Let's go down here. Prophase. The chromatin fibers change into chromosomes. So they're literally wrapping around those histones to make nucleosomes. They are condensed into those chromatid arms, which are attached by the centromere, and now they can be moved. And they're going to be moved to a metaphase plate, where the microtubules from the centrosome are now moving them to where they need to be. In anaphase, we're going to start to pull them to the poles. And what we mean by opposite poles is if you think back to those 2 cells, imagine one end of a cell and the other end of the cell. And telophase is where you actually now begin to finish the formation of 2 cells and 2 nuclei from what was once one cell. So the only way you can do that is take a balloon inside your mind's eye, right? Have 23 pair of chromosomes go to one end of the balloon, 23 pair of chromosomes go to the opposite end of the balloon, and then you put something right in the middle, and it's called an actin cytoskeleton, and it literally cinches itself, so you end up pinching off two small cells. They're actually pretty hard to find inside of a smear of actively dividing mitotic cells because they're only slightly smaller than all of the other cells around them. But you get the idea. We actually now have to divide these because we're going to go through what's called cytokinesis. And the division of the cell's cytoplasm into two will allow for that formation of that cleavage furrow I was just telling you about, okay? Where the actin cytoskeleton is what's responsible for now creating two daughter cells from one cell, okay? And once cytokinesis is complete, interphase begins on those two resulting cells. And this is the textbook version of what you see. This will be looking at inside the lab if you haven't already looked at it. Okay? So here's a cell interface. Look, you can't see dense chromosomes. You can see a nuclear envelope. In prophase, okay, what's going to happen is the nuclear envelope disappears and you can see chromosomes forming. You can see the individual chromatid arms, which is kind of amazing, right? And then the actual mitotic spindle is going to arrange all of the chromosomes here at the metaphase plate. And then as we move into anaphase, early and late anaphase, you're going to have the actual removing, pulling towards the poles here. You'll see they're not being pulled there. Towards late anaphase, you can see they've been pulled to the poles. And then as we move into telophase, probably a bad representation of it here. I think the reason why they chose it is you can actually see the actin furrow moving inward, but you can still see microtubules being shared here. You definitely do not see a nuclear envelope being reformed, but that's what you can see up here. And then once these relax, then interphase begins all again. Okay? So that's pretty much chapter 3. Because I think you guys can handle it, I'm going to introduce how meiosis is a little bit different than mitosis, right? So let's look over here. And mitosis would be a cell going along its normal processes. Then it goes through mitosis. Here's prophase, metaphase, anaphase, telophase, and two cells. Instead, what happens in meiosis when we need to make, if these are all diploid cells, 2N means diploid, and N means haploid. What happens is, in meiosis 1, I'm going to go ME1, meiosis 1, we have to recombine the genetic material. So you mix it up, okay? And you see this is what's called tetrad formation. You're going to take homologous chromosomes, you're going to break up the information, you're going to share it. This is what's going to cause a little bit of genetic diversity inside the gene pools, right? And what happens now is then you undergo metaphase 1 complete. Wham. And for males and females, it's different, but generally speaking, because this is an introductory textbook, they just put them all together and say, oh, look, figure it out later. So then what you're going to do is you're going to go through prophase 2, where you now separate the number of chromosomes that are attached to one another. Take a look here. You've got 1, 2 here, 1, 2 there, 1, 2 there. Now watch what happens. As you go through metaphase 2, you literally separate them from one another. Okay? And now you separate these further, so you end up with, if we started off with 2, 1, 2, 3, 4, 1, 2, 3, 4, look what you end up with down here. 1, 2, 3, 4, 1, 2, 3, 4. And these are the individual chromosomal arms. They originally came from mom or dad, but they'd been recombined here. So the purpose of meiosis 2 is to literally move away from a diploid to a haploid population, and then these two will fuse at fertilization. Okay? So what's the significance of all of this? Well, at the end of all of this, the beginning of chapter, what is it? Let's get rid of this first. Let's go here. The beginning of chapter 3, they say, oh, look, you're made up of cells, and cells are wonderful things. And inside lab, and as we continue throughout the semester, we're going to look at different types of cells. The average adult has nearly 100 trillion cells inside of their body. It's probably much more than that, but who cares? It doesn't matter. The important thing is that every single cell, if it's functioning, has a series of instructions inside its genes, right? Remember, that's the hereditary unit. And those are differentially transcribed and translated. And what happens is, when they're told to become something, they differentiate into these unique structures. So inside lab, you've seen the sperm cell, you've seen red blood cells, you've seen simple squamous epithelia or stratified cuboidal. That's the one I think I've given you as well. I've given you skeletal muscle and cardiac muscle so far, and I've given you different parts of a nerve cell. What I want you to realize is that each of these shapes is dependent upon how the proteins were built inside of individual cells to give them their unique function. That diversity will allow for organization of cells into more complex tissues and organs, which is hopefully what you've already seen, right? I mean, if you know the digestive system is made up of tips of your lips to the bottom of your anus, the purpose is that every single segment going through there will have a different population of cells doing a different job. That's a pretty good place to stop. So this is the end of chapter 3. Okay, I will see you guys in class if you're coming to class.